Good morning. You remember the previous lecture, we reached to the duodenum where there is a mixing of the bile with the pancreatic enzyme for the digestion of the food, which is reached to the duodenum. And we discussed the role of the gallbladder as a reservoir of the bile, and where the concentration is increased and the bile will be secreted under the control of cholecystokinin to the first part of the second part of the duodenum to form the vessel for the digestion of the fat and fat-soluble vitamin. So before we go to the what's happened in the small bowel, we'll discuss the type of electricity which is found to control the movement and the contractility of the gallbladder, of the small bowel and the stomach and the colon, and help the hormones and the reflexes and the CNS and the processing of digestion and absorption. So the first type of the electricity was called the basic electrical rhythm. There is a pacemaker like cells, the cagal cells. It's in the stomach and the small intestine and near the outer circular muscle where there is a mesenteric plexus, as you remember. And the function is to increase the muscle tension. In the colon, it is in the submucosal layer, so it is responsible for increase the muscle tension. The function is to coordinate the peristalsis and other motor activity. The function of the basic electrical rhythm is to coordinate the peristalsis and other motor activity. The other electrical activity, which is called the migratory motor complex, it is a cycle of motor activity migrate from stomach to distal ileum during fasting. They may clear the stomach and small intestine of the luminal contents in preparation for the next meal. So if you imagine that the middle lines is the times of the meal, so the migratory motor complex is help in the peristalsis to clean the gastrointestinal tract from the food and to prepare the duct for the second meal. So the basic electrical rhythm, it will control the peristalsis with other movement, and the migratory motor complex will work between the meal, to prepare the GIT for the next meal. So we will continue our journey in the small intestine. You know, we discussed the first part is the duodenum, the second is the jejunum, 40%, ileum is 60%. The small intestine length is 280 centimeter in vivo. You know, because in vitro it will be longer because there will be no contractility. The mucosa, as you see in the photo of the small intestine, contain a crypt and villi, like finger-like appearance, which is lined by the enterocyte and contain many of the enzymes which continue the last stage of digestion. Also in the intestinal mucosa, there is Brunner's gland and the goblet cells. As you see in the small intestine, the major part of the absorption is occur in the small intestine, and the macronutrient will reach by the digestion to the small units which help in the which can be absorbed. For example, glucose, galactose, fructose, amino acid, the peptide, the fatty acid. And there will be continued mixing of the intestinal content with the pancreatic bile and digestion completed, then the absorption of the product. So there is 9 liters of the fluid per day, 2 liters is the dietary source, and 7 liters is the GIT secretion. From the saliva to the gastric secretion to pancreatic secretion, bile, and the small intestine. So the content in the small bowel about 9 liters, 2 liters is the dietary source, and 7 liters is the GIT secretion. The absorption will occur in the small intestine when they are reaching to the ileocecal junction. It's only 1 to 2 liter will be passed to the large bowel. So most of these 9 liter will be absorbed and the remainder will be 1 to 2 liter which is reached to the colon. As I mentioned, intestinal mucosa contain the Brunner's gland, goblet cell, has a protective function, lubricant the food, hold immunoglobulin, the mucus secretion stimulated by the cholinergic stimuli and chemical and physical irritation. That means when there is a bowel food particles or there's maybe certain toxin or certain drugs, this may be stimulate more secretion. The types of small bowel intestinal motility. First, the peristalsis. As you see the upper photo, it is propel the intestinal content, which is called chyme, toward the large intestine. There is a contraction beyond the bolus, which is push the bolus toward the distal part of the small bowel. And so it's move the food particle from segment to segment, and you imagine it is like the toothpaste مثل معجون الأسنان. If you press on the tube, the content will be passed anteriorly. So there will be transmission of food particle from area to area. The segmentation contraction, it's move the chyme to and fro and increase the food exposure to the mucosa. The segmentation, from its names, it's move of the food in the one segment to go anterior and return back, and this will makes there is a time for the absorption because it is increase the time of the food to expose to the mucosa. The third type of contraction, it's isolation of one segment from the another. There will be isolation from one segment to another, and the last two contraction will be slow the transient time, while the peristalsis is shortening the transient time. As you see from this photo, this is the segmentation movement. You see in each segment, there it will be through and through of food particles to expose the food to the mucosa more. And there is a contraction between each segment to keep this process. Now imagine we are reaching to the ileocecal, to the end of the small bowel. And we review the lectures from the beginning for each macronutrient, the steps that we discussed before. For example, in case of the protein digestion, the digestion will be start in the stomach where there is a minor digestion by pepsin, and also there is a physical digestion by contraction of the muscle of the stomach, and then propelled of the food particles through the pylorus to the first part of the duodenum, and the second part where there will be the action of cholecystokinin, stimulate the pancreatic proenzyme secretion, the trypsinogen, the trypsin, and the help of the enterokinase. Also, there is activation of other enzymes, the trypsin and the chymotrypsin, to reach to the small bowel in the form of peptide and the amino acid, where it will be absorbed by the enterocyte. In case of the carbohydrate in the form of polysaccharide, starch, and sucrose and lactose, the digestion, if you remember, it will be started from the oral cavity by the mastication and also by the salivary gland amylase secretion. And then it will be transmitted through esophagus to the stomach by the gastric acid and the help of the contractility of the gastric wall. Then it will be transmitted to the first and second part of the duodenum with the help of the pancreatic amylase that will be changed to monosaccharide, glucose, galactose, and the fructose, and it will be absorbed in the small bowel. Of course, there is a disaccharides, for example, the lactose the milk sugar, it needs enzymes to make it to monosaccharide, which is called lactase, and this is present in the villus of the enterocyte, in the enterocyte of the small bowel. And they found that there is those people which are living in our area or in the surrounding area, which is called the Oriental population, have deficiency of lactase enzyme. For this reason, most of our people and the surrounding area have intolerance to milk, and this is which is called lactose intolerance because of the deficiency of lactase, which is present in the enterocyte. So deficiency of the enzyme cause diarrhea, bloating, flatulence. Why? Because the lactose will be maintained in the bowel human, so as a non-absorbable material, so it will be increase the osmotic pressure and increase the water transmission to the small bowel, and the bicolonic bacteria, it will be break down to produce the CO2 and the hydrogen. So those patients who are complaining from lactose intolerance, they will complain from osmotic diarrhea and from bloating and abdominal distension. So as I mentioned, the Oriental population, 70 to 90% of them have lactose intolerance. This information can help us in the medicine, in the management, when there is a patient which comes to you complaining of them diarrhea and distension after ingestion of milk. You can advise him to stop milk and you see the response if the patient is improved so that he has lactose intolerance. The third macronutrient is the fat. You know, the dietary fat contain triglyceride, cholesterol, and also this will be involved vitamin, fat-soluble vitamin, the A, K, E, and D. Digestion starts in the stomach by the chemical and mechanical digestion. There will be fat emulsification by motor activity to change to the fatty acid, where propelled to the duodenum, and the action of the cholecystokinin. It will be first help in the gallbladder contraction for the secretion of the bile, and second, it stimulates the pancreatic enzyme and its activation in the form of lipase and colipase, and there will be formation, as you remember, the micelle formation and the fat solubilization, and this will be transferred to the terminal ileum, where it will be absorbed in the form of monoglyceride and fatty acid. The carbohydrate absorption, a few notes about it. The transport of most of hexose is affected by the amount of sodium in the intestinal human. The sodium and the glucose share the same co-transporter. The glucose moves with the sodium and release it in the cells. So each molecule of the glucose has the ability of hold the sodium and enhance its transport through the intestinal mucosa. The water move into or out of the intestine until the osmotic pressure of the intestine equal to the plasma. The presence of osmotically active particle will be absorbed and the water is moved passively. The water will transmit to the media when it is high osmolarity. Try to make the osmolarity is stable. And from this physiological point of view, it is the base of the clinical application of magnesium sulfate, which is considered to be a poorly absorbed salt, and this retain their osmotic equivalent of water in the intestine. This magnesium sulfate is not absorbed, so it will be kept in the lumen of the bowel, and the water will be moved passively to the lumen, so there will be increase of the there will be softening of the stool, and this will be act as the osmotic equivalent of water and as a laxative effect. The other point for the electrolyte absorption, as we mentioned, the glucose will facilitate reabsorption of sodium. If the nutrient is not present, the sodium and the chloride will be absorbed by coupled activity of sodium hydrogen exchange and the chloride bicarbonate exchange. So there will be exchange of the electrolyte through the bowel wall. The chloride normally enter the enterocyte from the interstitial fluid in two ways. The first way via the sodium-potassium chloride co-transporter. Then the chloride will be secreted into the lumen via the cyclic AMP. The other pathway, the sodium-potassium ATPase and the sodium-glucose transporter. As we see in the next diagram, if you imagine this is the enterocyte cell and the left is the lumen of the bowel and the right the interstitial fluid, and you see that the chloride will be passed to the or absorbed in the two pathway. The first pathway by the control of the cyclic AMP, and the second pathway with the sodium chloride co-transporter enterocyte. So there is pathway for the chloride absorption, one with the cyclic AMP and other with the sodium exchange. And this is helpful in one of the clinical applications. In the cholera, you know, cholera is one of the disease which is caused by the organism, which is the Vibrio cholera, where the patient presented with profuse watery diarrhea, and which is called secretory diarrhea. Why this occur? Because the cholera bacilli toxin, it is increase the cyclic AMP, and this will increase the chloride secretion, and the sodium carrier decrease, so decrease sodium chloride absorption. While the second line for the chloride absorption, which is, as we mentioned, the sodium potassium ATPase and the sodium glucose co-transporter will not affect by this toxin. And this was the base of the manufacturing of which what's called the oral rehydration fluid. Oral rehydration fluid. It is type of the fluid which is contain the glucose and the sodium and the chloride, and it is manufacturing to give for the patient which complaining from diarrhea for fluid replacement. So the patient will lose with his diarrhea the sodium and the chloride, so they prepare a solution which is contain sodium chloride. And for the help the patient to absorb this sodium, they add glucose to this solution. So each glucose molecule that absorbed, it's carry a molecule of sodium and help to absorb it through the interstitial fluid, and this will help the patient to replace his fluid loss. To this slide, this lecture will be finished, and you have any questions, don't hesitate to ask. Thank you.